Hello everyone, I am Pekota Larisse, Assistant Professor from the Department of MLT. Today I'll be talking on the CRP test, the purpose, the method, the procedure and the interpretation. So the main aim of this test is to determine the C-reactive protein test by the agglutination method. So firstly, what is C-reactive protein? This C-reactive protein is a blood test marker for inflammation in the body, and this CRP is produced in the liver in response to inflammation. The other name for CRP, a high sensitivity C-reactive protein known as HS-CRP, and ultra sensitive C-reactive protein US-CRP. So this CRP is also performed to determine if a person has a problem linked to acute infection of inflammation. And the main purpose of this CRP is it is a slight agglutination test for the qualitative and semi-quantitative detection of C-reactive protein in human serum, where the latex particle coated with IgG anti-human CRP are agglutinated when mixed with samples containing CRP. So the latex particle coated with human anti-CRP are mixed with a patient's serum containing the C-reactive protein, which shows a visible agglutination within 2 minutes. So to perform this test, we will be requiring some materials such as the CRP kit. Here I have mentioned whatever is available in the CRP kit: DPC positive control, CRP antigen, NC negative control, and the mixing stick. Also, we are going to need normal saline, micropipettes, tips, and the serum. So there are, like I say, there are two methods: the qualitative method and the semi-quantitative method. But today I will demonstrate only on the qualitative latex agglutination method, or the CART test. So firstly, what we are going to do is that we before we start the practical, we are going to take out all the samples or all the reagents what we are going to need for the practicals, and keep it in a room temperature for 15 to 20 minutes before we start the practical. So the first thing we have to do is we have to dilute the patient's serum 1:5 with normal saline. Then we have to place a drop of positive control and negative control, and also the serum in the respective zone of the test slides. In the different zone, please don't get confused, not in one zone but in the different zone. So after adding all the PC and C and the serum on the respective zone, next we have to do is we have to add 1 drop of reagent, that is latex CRP antigen, in each respective zone, positive control, negative control, and the test. Next we have to mix well with the help of a mixing stick or an applicator stick, and then we have to rotate for 2 minutes. And after 2 minutes we are going to look for the agglutination. So how we are going to interpret is that here in the second image says that if there is an agglutination present, it means it is a positive test, and if there is no agglutination, that means it is considered as a negative test. So here in the test, since there is an agglutination showing in the test T, that is T, it is considered as a positive reaction, which indicates the presence of C-reactive protein at a significant and a detectable level. But here again in the NC, since the specimen they do not contain human CRP, so they do not cause any agglutination. So the main clinical significance of this CRP is that if there is a high level of CRP in the blood, It is a marker of inflammation, and also it can cause by a wide variety of conditions, like from infection up to the extent of cancer. So high CRP level can also indicate that there's an inflammation in the arteries of the heart, which can also mean a higher risk of the heart attack. And also if the findings of this CRP test can be very useful, and as a simple index of the disease activities and the in case of the treatment status. So this is all about the CRP test. It is a very simple method. This qualitative method or the card test is a very simple method. So I if you have any doubt, please feel free to clarify your doubt. Thank you.